Welcome. My name is Chris Ribbons, and I am the business manager at TEDx, the Endocrine Disruption Exchange. TEDx is a science-based nonprofit research institute. Our mission is to reduce the use and production of chemicals that interfere with healthy hormone function. We achieve this by identifying and answering pressing scientific questions and collaborating with partners to improve human and ecological health. I would like to welcome you to today's webinar titled, The Mammary Gland is a Sensitive Organ, Lessons Learned from UOG Chemical Mixtures. If you have a question for our speaker, please click on Q&A at the top or the bottom of your screen. We will be reading questions to our speaker after her presentation from the Q&A box, not the chat box, so please be sure that you type them in the Q&A box. Our host today is Kim Schultz, TEDx's Oil and Gas Program Coordinator. This webinar is part of an ongoing series addressing health effects associated with unconventional oil and gas development, where scientists present their research and answer some pressing questions. We hope that you will find the information useful, and we would welcome any comments or suggestions. You can contact us by going to our website at tedx.org. So now I will hand things over to you, Kim.
Thank you, Chris. I'd like to welcome everyone to our webinar today and introduce our speaker, Dr. Laura Vandenberg. Dr. Vandenberg earned her PhD from Tufts University School of Medicine in 2008. After postdoc positions at the Forsyth Institute at Harvard School of Dental Medicine and Tufts University, Dr. Vandenberg joined the faculty in the Department of Environmental Health Sciences at the University of Massachusetts Amherst in the fall of 2013. Dr. Vandenberg's laboratory research focuses on how low-level exposures to environmental endocrine disruptors can induce diseases, including infertility, metabolic syndrome, and breast cancer. Dr. Vandenberg is an author on more than 70 peer-reviewed papers and 9 book chapters. She has served on a number of U.S. and international expert panels to assess endocrine-disrupting chemicals, and regularly speaks with regulatory agencies around the world. Thank you so much, Dr. Vandenberg, for being on the webinar with us, and I would now like to turn it over to you.
Thanks, Kim. It's really great to be here, and I'm really excited to talk to you not only about UOG chemical mixtures, unconventional oil and gas chemical mixtures, but about my favorite organ, the mouse mammary gland. So as I get started here, I first want to introduce you to this very cool organ and explain to you why it has become a really useful tool for studying environmental chemical exposures, including these UOG chemical mixtures. So if we take a look at the mouse, and we're looking at the ventral surface of the mouse here, every mouse has 5 pairs of mammary glands that run the length of the animal's body on its tummy. And this is really helpful for mice that have litters because they can nurse more than 2 babies at a time, and they can nurse 10 babies at a time. And it's really useful also for scientists like me because one animal can produce a tremendous amount of tissue that can be used to evaluate the health of the mammary gland. So I'm going to give you just a little background on how the mammary gland develops in the mouse. And there are some similarities and differences with the human mammary gland, but the general features are the same. So there are 2 compartments of each one of those mammary glands. And the fatty compartment is shown here in this peach-colored oval. And the epithelial compartment is shown by the little black squiggly lines. And we think of the black squiggly lines, the ducts, as the functional part of the mammary gland. This is the part of the mammary gland that will eventually grow up to produce milk. And it's also the part of the mammary gland where tumors will form if an individual gets breast cancer. So when a mouse is born, it has this very small ductal epithelial tree. And a very large, relatively, compared to the ductal tree, a very large fat pad. In the center of each of those fat pads, there's a single lymph node. And this serves as a really nice landmarker to tell us how far along in development that mammary gland is. Now, as, excuse me, as the mouse grows, just from normal growth, the mammary gland ducts will grow at about the same rate until the female starts to produce estrogen when she starts to go through puberty. And as that happens, at the end of each of these ducts, there will form a bulb-like structure that's called a terminal end bud. And if we looked inside those bulbs, we would see epithelial cells that are very, very proliferative. They're dividing very rapidly. We would also see cells at the back of those terminal end buds that are dying. And they have to die because it's going from a solid structure to a hollow structure. We think of the mammary ducts as pipes because they will deliver milk. And these terminal end buds will chew their way through this fat pad and allow the mammary ducts to grow. Most of this happens during the pubertal period. And at the end of puberty, the mammary gland ducts will have fully filled the fat pad. Those terminal end buds will regress. And now we have what looks like a wintertime tree in that fat pad. And it will stay looking pretty much like this until there's another change in the female's life, notably pregnancy. At the onset of pregnancy, we'll see structures that look like little leaves growing on these trees. And those leaves are called alveolar buds. As the animal progresses through pregnancy, and pregnancy in a mouse is only 3 weeks long, so this is a process that occurs very rapidly, the entire mammary gland will be covered in these leaf-like structures. You won't even be able to see the branches. And that's because the animal will be preparing for making tons and tons of milk. After the mom weans her pups, those leaves will die. And we'll be left with a tree that looks pretty much like this, a wintertime tree with a few little leaves here and there. Now, the really great thing about the mammary gland, and what we've come to appreciate more and more with genetic tools and other more modern molecular techniques, is how different parts of this organ are affected by different hormones. And that this hormone and the role of hormones differs depending on the life stage of the female. So entirely different hormones are needed as the very initial mammary gland is forming in the embryo, from the kind of hormones that are needed to promote growth during puberty. And again, entirely different hormones that are needed to promote the massive expansion of those alveolar structures during pregnancy and lactation. My prior work has focused on a handful of chemicals, including the very well-studied BPA. And what we have shown is that animals exposed to BPA during early development have abnormal growth and development of the mammary gland at various stages of life, even when exposure does not continue into later life. This is showing you a study that's now more than 10 years old that has shown that the mouse mammary gland, and what you see in pictures A and B are those very, very early developing ductal trees. They're different in the BPA-exposed animals compared to controls. There's also changes to the fat pad that surrounds the mammary gland that we can see in the fetal gland. Other work, largely done in the lab of Dr. Ana Soto at Tufts School of Medicine, has shown that BPA can alter the appearance of the mammary gland. Again, these are animals exposed during prenatal life, and then looking at them at puberty, you can see here these terminal end buds, those bulb-like structures that are useful for proliferating and growing those ducts through the fat pad and chewing their way through that fat pad. So work from Dr. Soto's lab showed effects of BPA at puberty. Here you can very clearly see on the left-hand side that wintertime looking tree that I described, and on the right-hand side something that looks quite a bit different. So sort of maybe an early springtime tree in the BPA exposed animal, again, exposed early in life and now looking in adulthood. And by later adulthood, the BPA-exposed animal looks entirely different from the controls. And if you sent this BPA-exposed gland to a veterinary pathologist, they might guess that this is a gland from an early pregnant animal, and of course, it's not. And just a reminder that all of these effects take some time to manifest, that animals are exposed in early life, and we're seeing effects on the mammary gland that sometimes can take months to develop. Interestingly enough, we've also shown that the male mammary gland is sensitive to chemicals, including BPA, and other work that's ongoing in my lab has shown effects of other chemicals, including ethinyl estradiol, the common chemical found in pharmaceutical birth control pills, that male mice exposed in the womb to low doses of these chemicals also have abnormal looking mammary glands when they reach adulthood. BPA has been shown to increase precancerous lesions, including carcinoma in situ, in both mice and in rats. And also, a more recent study that came from Dr. Soto's lab at Tufts showed that some animals exposed to low doses of BPA during early development would actually develop carcinomas of the mammary gland. Finally, recent work from my group has evaluated animals that are exposed as adults during pregnancy and lactation, and has shown that the lactating mammary gland is also sensitive to estrogen exposures. In this case, looking at BPS exposed animals, and we saw that what you can see in the control picture here is this thick summer looking tree, lots of leaves on this tree. In fact, you can't even see those ducts anymore. Those are the structures that are actually producing milk, and you can kind of see through the BPS treated tree quite a bit more easily. So there's fewer of those milk producing structures. And when we actually sectioned these mammary glands, we can clearly see that what you should be looking at here on the bottom left picture, there's a lot of these very tightly compacted purple cells, and there's fewer of them in the BPS-treated animals. So all of this work that we've been doing for the last 10 or 15 years has really built up the mouse mammary gland as a good sort of sentinel, a good environmental sensor to be able to evaluate environmental chemicals, and in particular chemicals that interfere with the actions of hormones. And so this study that I'm going to tell you about, this new study, we used the mouse mammary gland to probe environmental chemical mixtures, and in particular chemicals that are used in unconventional oil and gas operations. In this study, the mice were generated in the lab of Dr. Susan Nagel at the University of Missouri in Columbia. And her lab treated female mice during pregnancy until they gave birth. And there were 4 different doses that were evaluated. And the female pups were collected, and mammary glands were collected specifically from them prior to the onset of puberty and then in early adulthood. And I know that Dr. Nagel has spoken on a TEDx webinar previously, and if you haven't heard one of her talks, I suggest that you go download it and listen to it now, because her group really did a lot of groundwork to evaluate the chemicals that would be used in the mixture that was administered to these mice. And in particular, they used cell-based assays to determine which of these chemicals had endocrine disrupting activities at one of 5, or sometimes more than one of 5 receptors. So you can see here the list of the chemicals on the left-hand side, and then specifically looking at their activity at estrogen receptor, androgen receptor, progesterone receptor, thyroid hormone receptor, and glucocorticoid receptor. And what Dr. Nagel's lab found was that these chemicals overwhelmingly had antagonistic activity, meaning they blocked the actions of the natural hormones that would be acting through each of those 5 receptors. And I don't want to sort of repeat too much what Dr. Nagel's group did, but her PhD student at the time, Chris Casotis, was the lead author on several papers that evaluated health endpoints in exposed male and female pups. I'm showing you here 2 endpoints that were measured in the exposed female pups. And you can see the 4 doses at the bottom referred to as mix 3, 30, 300, and 3000. And there were effects on the weight of the uterus that were different at these two different doses, the low and the high. And there were also effects on ovarian weight. So these are the same females that were evaluated in the mammary gland study that I'm going to tell you about. So we already knew that there was evidence that these females had been affected by these chemical exposures. Now, starting from looking at the effects of these UOG mixtures on the females at pre-puberty, so here you're looking at those, it looks pretty much exactly like the picture that I drew for you at the beginning with the little black tree. These are very small trees. There are no terminal end buds present in these trees. And while the animals exposed to all 4 of the mixture groups, as well as flutamide, which is a drug that blocks the actions of androgen and was included here to try to understand whether the mixtures were also blocking the actions of androgen blockers, here we see smaller ductal trees in all of the treatment groups compared to the controls, but these differences really were not statistically significant. By the time the animals reach postnatal day 85, so this is early adulthood, we can already see modest but significant changes in the amount of epithelium that is present in these animals. So shown in panel B is that at the mix 30 and 3000, there was a statistical trend and a significant increase at the mix 300 dose in the total amount of ducts found in these glands. So there's more epithelium here. We also looked at proliferation rates. So these are early adults, and early adults should have only about 1% of their cells dividing in the mammary epithelium at any particular time. We used Ki-67, which is the middle panel here, to measure proliferation. And what we saw is that at the low dose group, excuse me, mix 3, there's actually a high level of proliferation, a strikingly large increase in proliferation in that low dose group. This might be an example of a non-monotonic curve, although we only looked here at 3 of the treatment groups, so it's a little premature to really say that. We're also seeing a decrease in the percentage of cells that are apoptosing, which is marked by tunnel at the very top of the panel. And we saw no effect on the expression of estrogen receptor, which is the bottom panel of pictures. The super interesting thing that really caught my eye as a mammary gland biologist is that in these adults, we saw structures that looked like terminal end buds. They were at the ends of ducts, usually at the end of the mammary gland as far as it would progress into the fat pad. But this is an age where those TEB-like structures, those bulb-like structures, really should be gone. And we didn't observe them in the controls, consistent with what we would expect, that they should be gone. And yet we found them in the treated groups. And while our sample sizes were too small to reach statistical significance, you can see that there were quite an interesting number of these structures. And considering that I have probably looked at more than 1,000, probably more than 5,000 mammary glands from adult mice, this was the first time I had ever seen these abnormal structures in a healthy mouse mammary gland, an adult mouse mammary gland. We dissected those structures out and found that they actually aren't TEBs. They're instead highly proliferative lesions, epithelial lesions in the mammary gland, consistent with a precancerous lesion. So on the left-hand side of this image, I'm showing you 3 example lesions, and this is just a standard histology stain that a pathologist would use. In the middle is a marker of proliferation. You can see lots and lots of cells are labeled brown. And on the right-hand side is estrogen receptor expression. So when we characterize these, what we found is that those lesions are proliferating compared to areas outside of the lesions where we see less than 5% of cells are proliferating. Inside those lesions, we sometimes have 40 to 50% of those cells are proliferating. So they are dividing like crazy. And while in the tissue outside of those lesions, about 30% of cells are expressing estrogen receptor. Inside those lesions, about twice as many of those cells are expressing estrogen receptor. What this allows us to predict is that the estrogens produced in the bodies of those animals are likely going to drive further proliferation, further cell division inside of those abnormal lesions. So a few conclusions. One is that my favorite organ, the mouse mammary gland, I think is really a very sensitive organ and can be used to evaluate more than just chemicals that mimic estrogen, like BPA and BPS, but also chemicals that have a lot of other actions. I think that there is a lot of evidence that there are different developmental periods when the mammary gland is going to be sensitive to endocrine disruptors and also other environmental disruptions. And so I think it's important that we expand beyond just looking at fetal or embryonic exposures to also consider things like exposures to the mother. We know that disruptions can lead to disease, including cancers in rodents, and also abnormal function of the mammary gland like lactation. Of course, we don't know yet if those UOG mixtures lead to real overt disease or altered function. And this isn't the first study on mixtures, but it is certainly the first study on UOG mixtures that have evaluated the mammary gland. And clearly it is sensitive to these complex chemical mixtures. So we have a bunch of things to do, which is the great part about science. We're never done. One is that, and what we're working on right now in collaboration with Dr. Nagel's lab, is to evaluate mice that are kept for longer. So what are the long-term effects on the mammary gland after this early life UOG exposure? So do those terminal end bud-like structures go away, or do they progress to something worse? We want to understand if, because of the presence of those lesions in the mammary gland, we want to know if these animals are also more sensitive to a chemical carcinogen or to hormones. And I am particularly interested in whether mixtures, and in particular environmentally relevant UOG mixtures, will alter the function of the mammary gland. So does it prevent or improve the production of milk? Does it alter the composition of milk in these animals? So most of the work that I have shown you today came from Sarah Sapowski, my former student, and also Chris Kasodis, in collaboration with Susan Nagel's lab. This work was supported by the National Institute of Environmental Health Sciences, and I also have funding from the Breast Cancer and Environment Research Program, which works at the intersection of NIEHS and the National Cancer Institute. So I'm grateful to that funding, and I would be very happy to answer any questions that have come up during this presentation.
Well, I'd really like to thank you so much for your presentation. You did such a good job explaining things, and I really appreciate that. And so I think it's really interesting how all this animal work is coming out now and being published, and I really think it's invaluable to understanding how fracking really affects human health too. And so I'd love to start you off with a question on how do you think the doses you used in your study compared to human exposure levels?
It's a great question. And again, I think Dr. Nagel's lab has really been at the front of being able to address questions like that. Some of these chemicals have never been actually measured in the environment. So we don't know how much people are exposed to at or near fracking sites. But the two lowest dose groups, what we refer to as the mix 3 and the mix 30, were meant to replicate for as many chemicals as possible, excuse me, doses that would be found in drinking water in areas around fracking sites. The highest dose is meant to mimic the concentrations found in wastewater. So what would come out of the well after it's actually been sort of, the water's been inserted into the well, or pooling water around the areas of fracking sites. So probably the two lowest doses are the most relevant to humans living around those areas, and the highest dose is probably most relevant to the poor creatures that live around those areas.
Okay, thank you. Great, so some other questions. You did mention males before, and so I was wondering if this mixture could include chemicals that are androgen receptor antagonists, and if that would have any effects on male mammary glands.
Yeah, it's a really great question, and the answer is actually both entertaining and sort of forward-looking. The entertaining thing is that male mice don't have nipples. It's one of the few creatures that they don't have nipples, and so for a long time people assumed that meant that they had no mammary tissue at all. And the study that I showed you on BPA was one of the first where and it was totally by accident that we kind of found that those animals not only had mammary tissue, although they don't have nipples, that the mammary tissue is being affected by chemicals. So Dr. Nagel and I would very much like to evaluate males, but the mammary tissue is hard to collect. The female, you know, if you're even close, you're going to collect it. The male, it's this exceptionally tiny little bit of tissue. So we have to try again in order to be able to do that. But I suspect that anything that interferes with androgen receptor action will alter the mammary glands in those males. And what I would expect to see is actually larger mammary tissue in the animals exposed to the mixture compared to controls.
Okay, thanks. Let's see, some other questions we have here's a question. Is it possible to determine which chemical in the mixture is responsible for some of the effects you're seeing, like the mammary lesions, for example?
Yeah, so it's possible. Most of the chemicals that were evaluated here have never been evaluated even as single chemicals for their effects on the mammary gland. To study 23 individual chemicals, I would love if someone would give me the funding to do that, but it's unlikely to happen. And it's also incredibly likely that the effect is not because of a single chemical, but because of the interactions of these chemicals. There's actually a great body of work that's been done by Andreas Kortenkamp on this concept of what he calls something from nothing. So most of what he's done has been done in treated cells, but I think the idea would certainly apply to animals, that you can identify concentrations of individual chemicals that alone do nothing, and then mix them together and get an effect that you could not predict from looking at the individual chemicals. So I think it's a fabulous idea to try to find out who's the bad actor here when it comes to the mammary gland, but it would be a lot of work to actually sort of dissect that bad actor out.
Right, okay. And yeah, maybe there's not even one. That makes a lot of sense. Okay, and then I think our final question then would be, it's a two-part question. A lot of the chemicals in the mixture appear to have strong estrogen receptor antagonist properties, suggesting they might block the normal actions of the estrogens. So what effects might you expect on endpoints like lactation? And also, would you expect the chemical mixture to be protective against breast cancer like tamoxifen can be?
Great. So yes, blocking the actions of estrogen is, like tamoxifen, is used as in some cases, a prophylactic treatment to help prevent breast cancer in women who are at high risk. For example, women who have very dense breasts and a family history of breast cancer will sometimes be given tamoxifen, even though they don't have cancer. Tamoxifen is also used to treat estrogen-sensitive breast cancers once women do have them. That's not the same thing as asking what would happen during early development to exposures to these environmental chemicals. And the fact is, there's not a lot that's really been evaluated. I think people have assumed that something that mimics estrogen would be hazardous and something that blocks estrogen would be benign. But there isn't a lot of work on those kinds of chemicals. And so I think that that's one place where this work is really pushing into a novel area, is not to assume that blocking the actions of these receptors during development will have no effect on the mammary gland. Clearly that's not true. In terms of the function of the mammary gland, do we think that blocking the actions of estrogen might improve lactation? We have no idea, and it's a great question.
Okay, well, thank you so much, and I really look forward to your future research you mentioned on looking at the older animals and as they progress. I'm so excited about everything you're doing. Thank you so much, and thank you for presenting. And thank you for everyone for joining our webinar. And please stay tuned for more TEDx webinars in 2018. I'm going to send it back to Chris now to close this out.
Thanks, Kim. So thanks for listening today. We will be hosting more webinars in the future, and we'll send out emails to let you know about them. We also have recordings of previous calls and webinars on our website at TEDx.org, and would encourage you to listen to or watch the recordings and share them with others. Subscribe to our YouTube channel to receive notifications of new webinars. You can find us on YouTube by searching for the Endocrine Disruption Exchange. And be sure to follow us on Twitter. I would like to thank Dr. Vandenberg for taking the time to present her research, Kim, our host, and thank you for taking the time out of your day to watch. I hope that you have a wonderful day.
Thanks.